The plasma membrane of a cell is a network of lipids and proteins that forms the boundary between a cell's contents and the outside of the cell. It is also simply called the cell membrane. The main function of the plasma membrane is to protect the cell from its surrounding environment. It is semi-permeable and regulates the materials that enter and exit the cell. The cells of all living things have plasma membranes. The membrane is partially made up of molecules called phospholipids, which spontaneously arrange themselves into a double layer. Each phospholipid molecule has a head and two tails. The head loves water and the tails hate water. The water-hating tails are on the interior of the membrane, whereas the water-loving heads point outwards toward either the cytoplasm or the fluid that surrounds the cell. Within the plasma membrane, there are embedded molecules of cholesterol that are found alongside the phospholipids. Cholesterol molecules are amphipathic. Each molecule of cholesterol is composed of 4 fused carbon rings and a hydroxyl group. Within the plasma membrane, the hydroxyl group of cholesterol orients towards the polar phospholipids heads, while the carbon rings integrate between the tails. Cholesterol plays an important role in regulating the fluidity of the plasma membrane. At high temperatures, cholesterol rings interact with and partly immobilize the regions of the phospholipid fatty acid chains that are closest to the polar head groups. This decreases the fluidity of the membrane. At low temperatures, cholesterol increases the fluidity of the membrane by preventing the hydrocarbon chains of lipids from packing together and forming ordered crystal-like structures.
Proteins are the second major component of plasma membranes. There are 2 main categories of membrane proteins: peripheral and integral. Peripheral proteins are loosely attached to the surface of the plasma membrane. They can be found on either the cytoplasmic side or the extracellular side of the membrane. Integral proteins are the second major type of membrane proteins. Integral proteins have hydrophobic regions that anchors them to the tails of the phospholipids. Some integral proteins are partially inserted into the membrane, while others, known as transmembrane proteins, span the entire width of the membrane. Transmembrane proteins are further classified into channel and carrier proteins. Such proteins play an essential role in the transport of substance into and out of the cell. Channel proteins allow charged or large molecules to move down their concentration gradient from one side of the membrane to the other without using energy. Carrier proteins have a specific binding site for molecules that must be transported across the membrane. When bound, the protein changes shape, which allows the molecule to travel through the membrane and be released on the other side. Among the membrane proteins, a specific type of proteins exists known as glycoproteins. Glycoproteins are proteins with attached carbohydrate chains. These molecules are often located on the extracellular surface of the membrane and play key roles in cell recognition, signaling, and adhesion. Glycolipids, which are lipids with attached carbohydrate chains, are also present in the plasma membrane. Glycolipids' role is similar to glycoproteins. After the discovery of the cell membrane structure, the fluid mosaic model of the cell membrane was proposed in 1972. The term fluid mosaic model is used to describe the structure of the cell membrane because it captures 2 important characteristics: fluidity and diversity. The term fluid emphasizes the flexibility of the lipid bilayer, as individual phospholipid molecules can move laterally within the bilayer. The term mosaic refers to the diverse mosaic-like composition of the membrane. The membrane is composed of a variety of molecules, including phospholipids, proteins, carbohydrates, and cholesterol. The proteins are embedded in the lipid bilayer in a scattered or mosaic arrangement, contributing to the complexity of the membrane.